welcome to our HPL seminar today. So, I'm pleased to introduce our invited speaker who comes from McKeag Institute to join us today, Dr. Roman Krawetz. Dr. Krawetz received his PhD degree from the University of Western Ontario in 2006, and during his doctoral study, he worked with Dr. Greg Kelly and examined how stem cells from mice decide to differentiate into cells or tissues that make up the extra embryonic structures which can sustain the early embryo. After finishing his PhD, he flew to Calgary to start his postdoctoral training with Dr. Derek Rancourt here at the University of Calgary. His postdoctoral work demonstrated the usefulness of mouse and human embryonic stem cells for regenerative medicine approaches in bone and cartilage tissue engineering. His work generated a lot of interest, and he was awarded funding from CIHR and AIHS teams. This work also laid the foundation for him to transition into a faculty position in 2013 at the University of Calgary. Currently, Dr. Krawetz is an associate professor in the Department of Cell Biology and Anatomy and the Department of Surgery. He's also a full member of McKeag Institute for Bone and Joint Health. His lab focuses on stem cell biology and aims to further understand their role in the onset and pathogenesis of osteoarthritis, as well as in the cartilage regeneration. He has seen a lot of successes in this field, and in 2013 and 2018, he was awarded the Grace Glom Professorship in Arthritis Research, and in 2015 and 2019, he was awarded the Tier 2 Canada Research Chair in Bone and Joint Stem Cell Biology. Now, outside of work, Dr. Krawetz has a lot of interests, including going out to car shows or just relaxing at home, playing some video games. Without further ado, I'll pass the mic to Dr. Krawetz, and he will tell us more about the role of cell cycle activations in endogenous cartilage regeneration. Please.
The light went off. No, I think the battery might be dead. Wait. There we go. Okay. Or I just wasn't using it properly. That probably makes more sense. All right. Thank you for the invitation. Thank you for the introduction. It's been a couple of years since I've been here. And yes, I can see that my activities make me a perfect, my extracurricular activities fit in well with the HPL, but yeah, not really. Okay. Today I'm going to talk about some of the projects that we've had that was ongoing in the lab, and most of this work is actually was done by Christina Jablonski, who graduated last year. All right, so just a little bit about me. So it was already kind of said that I was interested in, one thing that wasn't said was the laser pointer is the top one? No. Center. Center, yeah. Thank you. So yeah, actually when I was doing my undergrad research, it was all in zebrafish. And then when I was switching over to PhD, it was looking at these embryonal carcinoma cells that are kind of like, they're a kind of cancerous stem cell in mice. But the one reason I want to point this out is just a show of hands. How many people have actually ever heard of a teratoma before? Okay? No? All right, so, but actually of interest is that probably at least one person in the audience right now has one growing inside them. So they're kind of a benign embryonal carcinoma that's actually it's fairly common in the population. You can see this one they can form like structures from all three germ layers. So this one has a little arm, some hair. They'll usually have teeth and bone, and that's the only. They're very rarely Well, does anybody want to show of hands who's had a chest X-ray in the last year? Okay, and did they? I guess you didn't have like teeth growing up in your abdomen somewhere. So usually that's where they're identified. So somebody goes in for a chest X-ray for cracked rib or something else, and they'll be like, "Oh yeah, it looks like you've got like some teeth in your body cavity," and then it's like a teratoma. But they're benign, so 9 times out of 10 they just leave them in there anyways because they don't grow much. They don't once they get to about like tennis ball or golf ball, they stop growing. So. Yeah.
That is the creepiest thing I've ever heard in the first 5 minutes of any talk.
All right. Well, if you ever see some teeth in an X-ray, then you pretty much you'll remember it from you heard it here first. All right. So the lab, we're interested in kind of two stem cells in 2 different ways. So we're interested in kind of endogenous stem cells, so those stem cells that are already in our tissues right now. All of us walking around in this room have active stem cells, and if you didn't, you wouldn't be walking around. People heard of or seen the disease progeria, so those young children that look really really old, and it's like they expire very young in life, and one of the thoughts behind why that happens is because of stem cell exhaustion. So their stem cells just exhaust and they're no longer functional. So then your tissues break down a lot faster. But we're also interested in potentially looking at if we can understand these endogenous stem cells, then we can use that to develop new therapies using stem cells. We're also the disease that we're focused on is osteoarthritis. So making the diagnosis of OA more efficient. And then also, using some of these stem cell therapies and or pharmaceuticals that drive stem cells as a treatment for OA. All right, so I won't, this vicious cycle of stem cells, or vicious cycle of osteoarthritis, I'll kind of just skip over this, but I'm sure most people in the room know that it's like usually it was historically considered a wear and tear disease or a disease of old age. And in the last, 15, 20 years, that paradigm's been broken, where it's a disease with multiple risk factors and multiple health outcomes and a whole bunch of different pathways in the middle, and it's very heterogeneous, and it's actually that makes it very difficult to diagnose and understand as progression. All right, so a couple of the projects that we have in the lab, so we look at exogenous repair, so that's taking stem cells from one source or one animal or one person and delivering them to another animal or another model, just to see if we can trigger repair by delivering stem cells. I'm not going to talk about that. I'm not going to talk about inflammation too much today. Another project we have is when this started with tannin, is this idea of what lubricant's doing in this body, doing in the body, and it looks to be more than just lubrication. And what I'll talk today about is endogenous repair. So can we get our own stem cells to kind of get off their butts and get to work and fix our bodies, or especially our cartilage? And what is the role of inflammation in this repair or lack thereof? All right, so articular cartilage. Everybody's got it, and you really don't know that you're not supposed to know it's there until it doesn't work anymore. It has no nerves, has no blood supplies. You don't feel it every time you take a step. But then, when it's lost and when that bone that's underneath it is highly vascularized, highly innervated, and what's been told to me that people that have full thickness cartilage lost, it's like every time you step, it's like having a shot of like tooth pain when you've got a cavity or something. You take a cold drink of water, you feel it. That's what I've been told it's analogous to, and based on what my left knee feels like most mornings, I'm sure I'll be there sooner than later. Okay. All right, so why do people with cartilage damage go on to get osteoarthritis? So as early as documented as early as the mid-1700s by William Hunter, it's this concept that basically mammals don't repair cartilage. All right, so if you have a small cartilage injury in the joint, that'll just spread over time until it gets down to the full thickness of the cartilage is lost, and then you kind of go on to develop osteoarthritis. Now there are some notable exceptions to this, one of them being rabbits. Rabbits are just a strange kind of one-off animal in general, so they kind of do weird things, and one of the things they can do is, in some cases, they can actually regenerate cartilage tissues. And then in mice, there's Some interesting mice models that have been shown to regenerate cartilage, and that's what we'll be talking about today, but still the mechanisms of how these mice can do this is still kind of elusive. So these pictures on the side here, you're looking at, this is mouse articular cartilage stained with saffron O. This red stain at the top of the cartilage, that's it's staining positive for the proteoglycans, so that what gives the cartilage its ability to resist compressive forces by retaining water. And if you damage that so we've got two mouse strains here. So this is C57 Black 6 mouse. This is kind of your generic lab mouse. Very similar to humans, C57 Black 6 mice do not repair cartilage injuries. If you poke C57 Black 6 mouse cartilage, that defect will just spread and spread and spread. And that's what we're kind of showing here. Over time, it's like this attempt at a repair response. You get some fibrocartilage or fibrotic tissue without cells, and then over the long term, it just degrades and you get right down to the bone. These MRL super healer mice, which are, it's a funny story. It's mice are small, average here, 25 grams or so. That makes basically almost everybody in the orthopedic realm hate mice because they're really small. So people, if you're going to use a rodent model in orthopedics, you typically go for a rat. But the problem with rats is that you can't really do transgenic modifications on them. You can nowadays, but it's still really expensive. So about 20 years ago, a group of people wanted to selectively breed a really big mouse. So they wanted to get a mouse that was the size of a rat, so you could do transgenics on kind of like a rat mouse. And unfortunately, through all that selective breeding, they got a mouse that's about twice as big as a regular mouse, so about half as big as, or less than half the size of a rat, but twice as big as a mouse. But that mouse gets all sorts of autoimmune diseases and cancer, so then nobody wanted to use it except for people studying autoimmune diseases and cancer, which they did for a long time. And eventually, when those groups, like the autoimmune group, started to look at transgenics in this MRL mouse, which stands for Murphy's-Roth's large, if you poke the ears, so typically when you're genetically modifying mice, you do an ear punch so you can track them. So this one's got this genotype, this one's got this genotype. And when they do that and let the mice run around in a cage for a month, the ear punches all disappear. So much like articular cartilage in the joint, auricular cartilage in your ear does not heal. Same thing in mice. And so, but these mice were able to heal those auricular cartilage defects. And then groups went and saw, oh, look, can they also heal the articular cartilage defects? And lo and behold, over time, they can. So they can regenerate that kind of full thickness articular cartilage after injury. And then, so like over the last, so that was kind of figured out, that was demonstrated in like 1996, 1998 was the first time. And over the last kind of almost like 20 years, people have been trying to figure out how these mice repair, these MRL mice, how they repair their tissues. And so one of the things that people looked into, the first thing they looked into was because these mice have autoimmune disorders. We know that most autoimmunity is driven by inflammation. So does inflammation play a role? And it's basically, long story short, it's entirely possible. So there was a different inflammatory response to injury in MRL mice versus a C57 mice, but it's maybe not what you would think. MRL mice have a very, very high acute inflammatory response. So they get injured, they have this huge pro-inflammatory response that trumps like a C57 by multiple times, but it disappears within 3 to 4 days. So you get this really potent burst of inflammation, turns off. C57 Black 6 and most other mice, and actually people, how they respond to injury is you slowly ramp up this pro-inflammatory response over about a week, and then in the best case scenario, that disappears after about 2 weeks, but in chronic inflammation, it just stays. And so that's what you kind of see in C57 Black 6 mice in people, that you ramp up slowly an inflammatory response and it just stays. MRL, it's like it comes on really strong in about 3 days and then it's kind of gone. So what we did in the lab was we took the immune system from an MRL mouse and put it into a C57 Black 6 mouse, and took the immune system from a C57 Black 6 mouse and put it in an MRL mouse. And that's, a lot of people, mice might not be the best orthopedic model, but you can do really funky experiments like this that make it worthwhile to study mice. So what we found is that even if you give a C57 Black 6 mouse, it's called the chimera, the immune system of an MRL mouse. This is a cartilage grading score, with high numbers being better. They don't repair cartilage, right? So they even a C57 Black 6 mouse with an MRL immune system doesn't work. And if you actually give an MRL mouse the C57 Black 6 immune system, it doesn't matter. The MRL still repairs. So this really suggests that the immune system, and like this kind of inflammatory response, it may play a role, but it's not playing the major role in this kind of endogenous cartilage repair. So then, since we're a stem cell lab, obviously we wanted to look into stem cells. Just a little one slide on kind of prep, their background would be that stem cells, there's many different types of stem cells, like embryonic stem cells, induced pluripotent stem cells. But the stem cells that all of us have in our bodies right now are termed adult stem cells. So you have stem cells within every one of your tissues, including your brain, right at this moment, that are doing whatever they need to do to keep you going. The one that's debated, the tissue that is debated is cartilage. So if you ask 10 stem cell biologists, does cartilage have stem cells? 5 will say yes and 5 will say no, but regardless, we all know that cartilage really doesn't repair itself, so I'm kind of on the no side of that camp. But anyways, these are very rare cells. So you're talking about maybe of every 10,000 to 250,000 cells in your body, not including like red blood cells, that you might have a stem cell. These cells have been shown to have immunomodulatory properties, so they can regulate inflammation. And they've also been shown to be able to differentiate, so they can turn into other cell types. And so we were curious, do these cells play a role in cartilage repair? So since we don't really, our lab hasn't been able to find a stem cell population in cartilage, but the field does completely agree that there's actually a stem cell population in your synovium. And your synovium is the tissue that lines the inside of your joint, analogous to like the lining of a balloon, right? So it just kind of lines the entire inside of your joint. And for those of you that kind of like kind of a tissue that synovium is pretty analogous to is like the inside of your mouth. So the inside of your mouth is actually pretty similar to the inside of your joint. Tissue-wise. And so that in just the same way the inside of your mouth is full of like stem cells, so is the inside of that lining of your joint. So what we did was we isolated stem cells from the synovium of the MRL mice, so the mice that can repair, and the C57 Black 6, the mice that can't repair. We infected those stem cells with green fluorescent protein, so now all the stem cells are green. And then we injected them back into only C57 Black 6 mice because they can't heal. And we gave those C57 Black 6 mice all a cartilage injury. And so this is all these immunos here. This is the blue is the DAPI. And this little area here is the defect. So, and this is C57 Black 6 mice that have been injected with C57 Black 6 MSCs. And we don't see really any GFP in the defect area. But if we take MRL MSCs, and that's the defect area, you can see that you've got this green there. And if you get rid of the DAPI and go a higher magnification, you can see the defect area is pretty much full of these MRL stem cells. So those stem cells migrate to the joint, and we actually get an increased cartilage repair, suggesting that these stem cells are playing a role in this ability of these animals to endogenously repair. Now, the interesting thing is if you go back 4 weeks later, so in the C57 Black 6 mice, you really still don't see stem cells, you see 2. There's 2 there. But even though this was all green and all full of stem cells, when we go back a couple weeks later, there is there's no green cells. And so what that means, by 4 weeks, we already have that new cartilage layer. The cartilage is already starting to regenerate, but none of that new cartilage was actually made by the stem cells. So the stem cells went to the defect, they've done something, and then they've either migrated out or they've died. So that we kind of took a step back and then tried to figure out what our next line of attack was going to be, and went back to this kind of what are some of the what could be some of the potential mechanisms for why these stem cells are acting different and we're getting new cartilage, but the stem cells aren't making new cartilage. So I already kind of talked about that in the MRL mice, inflammation is differentially regulated. In the MRL mice, it's also been found that like these pro-healing molecules like TGF-beta are differentially regulated. But the one that we cued on was that a group in 2010 showed that if you knock out P21, which is a cell cycle, it's a gene that regulates cell cycle or the ability of cells to divide, you can actually get, you get cancer and autoimmune disorders, but you also get that ear regenerating ability. So they didn't check it. They didn't check in the joint, but they found that if the P21 knockout mice, if you punch the ear will close. So just what P21 actually does is this is every time one of your cells is going to divide, it goes through these different checkpoints, and that P21. So these different checkpoints are driven by cyclins. So the expression of cyclins allows cells to go from checkpoint to checkpoint, and P21 just inhibits these cyclins. So it just stops cells from moving from one checkpoint to another. And if you delete P21, all of your cells, like you get an accumulation of cells in this G2M. So they just kind of fly through the cell cycle and accumulate here. So you get increased proliferation. And that there had been some evidence in the past that maybe P21 could be playing a role in kind of cartilage repair or chondrogenesis. So we, in our lab a couple years ago, we actually found that in humans, so patients with osteoarthritis, if we look at their synovial stem cells, so their stem cells in their joints, they have increased P21 expression, increased inflammation, and those stem cells actually have decreased ability to turn into cartilage. This is in patients, so we don't know if it's a direct link, but it was circumstantial evidence. Another group, this was actually Farsh-Gilak's group. They deleted P21 in induced pluripotent stem cells. And when they did that, those induced pluripotent stem cells actually had an increased ability to become cartilage. So again, and then that paper from 2010 that if you delete P21 in mice, they can repair the ear injury. And this is from our lab, not from that paper, but it's just you can see that you punch a hole, and over 4 weeks, the hole disappears. So what we wanted, the first step we were just going to take was we wanted to see if P21 knockout mice could actually regenerate articular cartilage. So we have C57 black 6. These are mice that can't regenerate. These are the P21 knockouts that they can regenerate the ear. And then we make a defect, full thickness cartilage defect, right in the femoral groove. It punctures all the way down to the subchondral bone, and then we follow these up at 1 hour, 3 days, 1 week, 2 weeks, and 4 weeks. And in this study, we followed this up through standard histology using that saffron O. And we also followed that up looking at 3D X-ray microscopy, which is, it's somewhat analogous to CT, that it's designed to look at bone. But this wonderful, expensive, heavy piece of technology, also you can modulate the energy very tightly in this system, and you can actually do very low energy. And so what low X-ray energy allows you to do is visualize cartilage. And so what you can do in the 3D X-ray microscope is not just visualize the bone, but also visualize the cartilage at the same time without adding any contrast, which is, there's to my knowledge, this is the only type of methodology on the face of the earth that can do this at the moment. At very high resolution. But the drawback of this is that it is, it's ex vivo. You cannot do this in living animals because of it's basically like a microwave and just ashes things after a while. So All right, so if we, first and foremost, looking at histology, P21 mice regenerate articular cartilage. So C57 black 6, that red surface, you make injury in it, and over time it disappears. P21 mice, you make an injury, you get this burst of kind of repair, repair tissue, and then over time it kind of gets back to looking like a articular cartilage surface. And if you grade that, you can see that it's, yeah, there's an increase in cartilage repair. Now these next couple slides, all the pictures are going to be like this. So this is the X-radia, or the X-ray microscopy. And so the white is the bone. And then this green box just shows the area where the cartilage injury was made. And then this little circle is that region of interest. The green is we've thresholded out the cartilage, and the white is actually the bone underneath. So we're looking at the cartilage and the bone at the same time. So C57 black 6 mice, as I said, they don't repair that. They have that fiber cartilage repair, right? So it's just that degrades over time. And the P21s have that articular cartilage repair. The one limitation of this is that we can't tell the difference between fiber cartilage and articular cartilage, and that's why we have to rely on the histology to see what type of tissue formed. But what we can see is that, yep, so if we, a normal uninjured joint, you have bone underneath and green cartilage on top. You make a hole, you damage the cartilage, you damage the bone, and then within 3 days, you can already see that the mice heal bone really, really quickly. You can see that the subchondral bone is already filled in both mouse strains. And what's interesting is you can see that it looks like so this fiber cartilage and this articular cartilage looks to be healing from the outside in. And then if we go over further time points, you can see that's actually the case. And by 3 to 4 weeks, you've got a complete fiber cartilage patch and a near complete articular cartilage patch in these animals. And then if we quantify the thickness, this is thickness compared to the uninjured animal. The P21s, we can see they've got this increased cartilage thickness. And if we go back to the histology, we think this is what's actually giving us that increased cartilage thickness, that little bit of cartilage that grows into the damaged bone, and then just kind of that then remodels. Okay. So summary of this part is that we do see the P21 knockout mice actually can spontaneously regenerate their articular cartilage surface, and it appears that they've got that thicker cartilage that then remodels into normally thick cartilage. What we were interested in after this first step is what cells. All right, so how do you know what cells are actually doing it? Because if you can find out what cells are doing it in this P21 mouse, and we have the same cells, then we might be able to take advantage of that and either modify human cells to do the same job, or just potentially give more of those cells to get an effect. So we thought pretty long and hard about how we were actually going to do this, and we decided to do it genetically. And so using lineage tracing. And so what I mean by lineage tracing is that we wanted to fluorescently label the stem cells that are already in the mouse. So not add any more, not actually really have to do anything to the mouse, but just we can fluorescently label the existing stem cells in the body, make an injury, and see if those stem cells are the ones that actually respond to that injury and repair the defect. And so how you do lineage tracing is that you need to pick a promoter, a gene promoter. And the promoter that we picked is called this paired homeobox 1. And so why would you pick paired homeobox 1? And there's a whole bunch of reasons, like, yes, all the cartilaginous structures in your body during development are made by paired or PRX1 cells. But more important for us was that if so in your body, articular cartilage or ear cartilage or nose cartilage doesn't regenerate. But what's interesting is that anytime you break a bone, that fracture is typically, like if it's a fracture that's healed by endochondral ossification, which most are, long bones, you get a cartilaginous callus. And in mice and in humans, that cartilaginous callus is actually 100%, is near 100% derived from stem cells. It's a transient structure that doesn't stay around for long, but while it's there, it's in mice, we know that that cartilaginous callus is completely made of these PRX stem cells. So we know that in the body, PRX stem cells can become kind of articular-like cartilage in the adult, which not very many other cells can do. So what we do is we take a PRX. This is so we got a PRX1 mouse. So it's called PRX CRE-ERT2 GFP. What this means is that any cell that's expressing PRX, so that's our stem cells, are going to also be expressing green fluorescent protein. But then, because if a stem cell differentiates, it won't be PRX positive anymore. So then it won't be GFP positive anymore. So to make it so that we can follow it even after it's differentiated, we bred that mouse to a mouse that has a TD tomato gene, which is basically red fluorescent protein, just bright red. So undifferentiated stem cells are green and red. And then when they differentiate, they lose the green and they just stay red. Right? So undifferentiated stem cells, green plus red, which equals yellow. And when those stem cells differentiate, let's say they become new cartilage or new bone or whatever, they will remain red. So experimental plan for this is that we take the P21 knockout mouse, the C57 knockout mice that have these transgenes or these genes that we've inserted in them. We inject them with tamoxifen that activates the genes. And then now the cells are green or red. And then we just follow up kind of on the same time points. We did one later time point as well after the cartilage injury. And so we looked at immunofluorescence so we can find those stem cells. And that's just regular histology so we can grade how well the mice actually repair cartilage. And Luminex assay because we wanted to see not only if the stem cells are playing a role, but also get back to this inflammation. So if inflammation is playing a role. All right, so because we genetically modified these animals, so we've messed around with their genomes, we've added genes that aren't in a normal mouse. We wanted to make sure that all those genetic alterations didn't mess up the P21 knockout ability to regenerate cartilage. P21, so higher is better, so P21 might still demonstrate better articular cartilage regeneration. And then these are back to the immunos. This is immunos of the femoral groove, so where we did the damage. And then so the blue is DAPI, that's the nuclei. The C here is cartilage, patella, synovium, subchondral bone. And then the undifferentiated PRX cells are in green, and the red differentiated PRX cells or cells that used to be stem cells are in red. In a non-injured joint, either C57 or P21 knockout, you don't have these stem cells just aren't sitting in your cartilage, right? So it starts to validate that point that we don't see green positive cells in the cartilage, we don't see red cells in the cartilage. You see red and green cells in the synovium, which is what we would expect because we know there's stem cells there, but they're not just hanging out in the cartilage. Higher magnification, you can see the same thing. But when we make that injury, and then 1 week later, so C57 on this, P21 on the bottom, is that you get infiltration of stem cells into that cartilage defect, right? So you get stem cells that are migrating into this defect area, but the vast majority of them are still green. So that means they're undifferentiated. So these are undifferentiated stem cells migrating into the defect. Interestingly enough, we actually didn't see that in P21 knockout mice. We saw very few stem cells migrate into the defect. We saw more stem cells in the subchondral bone. If you go to, I skipped a couple time points just for time, but if you go to 4 weeks, so 4 weeks is when we're already seeing that cartilage regeneration. In the C57 black 6 mice, we see very few stem cells in the defect. They're kind of around the defect, but not in the defect. But in the P21 mice, what was interesting is we consistently saw stem cells bordering the defect. So these undifferentiated stem cells were now bordering the defect. And so one of the issues clinically, so in patients when they're trying to do cartilage repair or like an osteochondral replacement in a patient with a focal cartilage defect, they take a piece of cartilage from like a normal part of the joint or from like a donor and transplant it into the part that's damaged. One of the things that actually why those treatments fail is because there's either no or poor integration of the new cartilage to the old cartilage. And so that's what we kind of think that these cells might be playing a role in kind of at integrating the new cartilage and old cartilage together. We're still working on proving that, but that's what it kind of looks like. And if you go 8 weeks, so the C57s, again, you got some, a couple green cells here, some green cells there, but it's the P21s that they've regenerated that cartilage surface, and you see very few stem cells, and importantly, none of that, like very little or almost none of that cartilage surface is actually derived from stem cells. So even though we're getting a new cartilage surface, it's not derived from the PRX positive stem cells. And so if it's not the new stem cells, so maybe stem cells are playing a role, but it's not like the stem cells aren't building new cartilage. So we ran the blood from these animals, and then we kind of, in these ones that are kind of yellowy, that means these are all pro-inflammatory cytokines, except for IL-10, which is anti-inflammatory. But if it's kind of yellow, then that means it's higher in P21 than C57, and if it's green, it's higher in C57 than it was in P21. And similar to what people saw in the MRL mice, we get a higher inflammatory response in the P21s that disappears, and then we get a chronic inflammatory response in the C57s that goes to the end of when we looked. The one thing that is interesting, and we're going to come back to, is that CCL2, which is actually a cytokine that's been implicated in osteoarthritis, osteoarthritic pain, and basically cartilage degeneration, was actually up in P21s at the end. And you can see it kind of bucks the whole trend. And that was, we weren't expecting that at all. So we're going to get into looking at that in a moment. All right, so if we summarize this part here, P21 knockout mice, we get that hyaline-like cartilage, so versus fiber-like cartilage, so they can repair the articular cartilage injuries. We quantified the PRX cells. So PRX cells. We quantified other MSCs. We quantified macrophages, and because P21 is involved in proliferating cells, we also quantified the proliferating cells, and just didn't show all that because there would have been a lot of other pictures of immunos. And when you do the quantification, there's no difference in total numbers. There's a difference in kind of localization, as I mentioned, but there's no difference in total numbers of stem cells or macrophages. And then if you look at the inflammatory response, you get this late/chronic response in the C57s, and you get this early acute response in the P21s. So the last part of the talk will be trying to figure out what we saw with that CCL2 in the P21 knockout mice. So as I was saying, there's a couple papers that have come out in the last couple of years linking CCL2, which stands, well, actually, it's like C-type chemokine ligand 2 or something like that is what it stands for. It's not a very creative name. And CCR2 is basically C-type chemokine receptor 2. So this is the ligand, this is the receptor for that ligand. So both the receptor and the ligand have been implicated in, some papers have said osteoarthritis, some papers have said pain. Most papers agree pain. Not all papers agree that it's actually related to like cartilage degeneration, but there's a lot of other groups actually studying this right now in osteoarthritis. All right, so what we did is we looked at knockout mice for CCL2, knockout mice for CCR2, and then also did the double knockout, so you remove the ligand and the receptor. And so you see here we've got the CCL2 knockout mice, the R2 knockout mice, the double knockout mice, and we did the same full thickness cartilage injury. We also did the OA, which I'm not going to show. and did immunohistochemistry to find, proliferating cells, mesenchymal stem cells, macrophages. And then we actually also isolated the stem cells from these mice to look at their differentiation ability. So the long and the short of it is that if you knock out CCL2, which we were surprised again, that you don't get cartilage regeneration. So you knock out the ligand, no cartilage regeneration. You knock out both, no cartilage regeneration. You knock out just the receptor, you get cartilage regeneration. So it's not as perfect or pretty as the P21 mice or the MRL mice, but you do get statistically improved cartilage regeneration. And so what this says is that CCL2 is actually required for endogenous cartilage repair. And if you get rid of it, even if you, so the CCR2, the receptor is somehow inhibiting cartilage repair, so you get rid of it, you get regeneration. You inhibit the ligand, no regeneration. And if you inhibit the ligand and the receptor, no regeneration, saying that CCL2 is absolutely required for cartilage regeneration, but is not signaling through its main ligand, or main receptor, which is the CCR2. Sorry if that's a little convoluted, but yes. So even though people have been implicating this in cartilage degeneration and pain, it does look like it's required for cartilage regeneration in mice. And then the pages and pages of this, but I'm not going to show it because it would be very monotonous. But we quantified all of the quantified stem cells, quantified macrophages, both in the synovium, the cartilage defect, and the subchondral bone. But what we found, the big take home, is that in the CCR2 knockout animals, we found fewer cells actually in that cartilage defect area. So it's a similar kind of what we're seeing to the P21, where we might get cells bordering the defect, but we don't actually get cells in the defect. So there's something about keeping the cells out of actually the cartilage injury, or keeping these stem cells and macrophages out of the injury site, that somehow promotes endogenous repair of cartilage. And then lastly, if we took the stem cells out of the bone marrow from all these different mice, and we found that the CCR2, so when you knocked out the receptor, they had increased ability to become cartilage. And so this was a little confusing to us, and we're still working on this one. But so modifying this inflammatory pathway also modifies the ability of the stem cells to actually potentially contribute to repair, even though they're not really contributing to, they're not becoming new cartilage. It somehow has something to do with their ability to become new cartilage in a dish. And what we think is going on here is that we've backed this up with some proteomic experiments. Is what we think the stem cells are doing is that they border the defect and they secrete cartilage extracellular matrix proteins. So they don't become the new cartilage, but they're secreting the extracellular matrix proteins like collagen 2, aggrecan, biglycan, and we think that they're actually just secreting that into the injury site, and then new chondrocytes are just migrating into it. We still have to prove that, but so far it looks to be the most likely hypothesis. All right, so that CCL2 is necessary for cartilage regeneration, CCR2 is inhibitory, and that this CCL2 mediated response, it must be signaling through a different receptor. So we don't know what that receptor is, but there are a couple choices. One thing I didn't show is that if we also deleted the whole pathway, that CCL2 and CCR2, the mice were partially protected, but not completely protected from osteoarthritis. All right, so kind of trying to wrap this all up in one slide is that we do, in a C57 black 6 wild type, and similar to a person, when you get a cartilage injury, it repairs with a fiber-like cartilage patch, like a scar. That scar, best case scenario in a human, will last about 2 years. A lot of times won't even last a year. And then it degrades down to the bone, and then you've got full thickness cartilage lesions. In the P21 knockout mice, we see that we get a repair of that hyaline-like cartilage. And even though we don't see any difference in the number of stem cells or immune cells, macrophages, in between those two injuries, we think it's how they're being localized, like around the defect, and maybe what they're secreting into the defect that is leading to that endogenous cartilage repair. And then just wanted to, the people black and underlined are the ones that contributed to this project, and the vast majority of the work was done by Christina Jablonski, who graduated last year and is now doing an industrial postdoc in zoitis in the States. And thank you very much. Yeah, a lot of information.
Well, always hold that for a question.
What's powering all of this? Since that cartilage is supposed to be avascular, so is this all basically powered by diffusion of nutrients?
Yeah, so that's in theory, like, or in practice, I guess you would want to say yes. And the reason that we think it's probably like that is because we know that, strangely enough, even though stem cells are like a fairly active cell type, they're not, they do better under like low oxygen conditions. So you can't, they will work fine in a wide range of nutrient conditions, but you can't starve them completely. But you can actually, about 1 to 2% oxygen, if not, like, you can probably get them down to about 0.5% oxygen, they'll actually perform better. So that it is entirely possible that they don't really require too much to do what they're doing here.
Has anyone ever looked at the cartilage repair process during growth and development?
Yeah, so
what happens when you give the insult to them when they're growing?
So fetal, that's actually been done in, it's been done with mice, and it's been done with other animals, but importantly, it's also been done with recovered human fetal tissue. And yes, like, if you damage the cartilage of a developing human fetus, it'll repair scar-free. So, like,
what about, like, not a developing fetus, but like a child, or like young black 6?
Yeah. So yeah, there's also, so short answer is yes, there is, like, the younger the mouse, the more capacity it has for endogenous repair. But there's actually there's this weird thing with timing that, like, we don't do that, but there are a number of big labs around the world that are doing that, and that certain tissues, what was it? There's one tissue that, the heart. So if you slice the mouse's heart open at day 1, like when it's, like, basically it's been born on the first day of its post-uteral life, that heart will stitch, like in almost any mouse, that heart will stitch together scar-free. If you do it on day 2, it won't. And that there's other tissues that, so another one that's kind of getting more traction right now is called the digit tip, right? So mice have claws similar, probably just like, they kind of look like little cat claws, but just really smaller. And you do see that, like, mice that are, I think it's like the first 5 days. It's something like the first 5 days of a mouse, like, post-uteral life, you can cut off, like, pretty much most of that first digit, and it'll regenerate. And then as the mouse gets older, you can cut off, like, less and less, and it'll still have the ability to regenerate. I think it's like day 14 or day 15 of life is, like, the last time you can cut off anything with it just not scarring. So, like, yeah, and that it would be the couple. There's not a lot of similar papers on articular cartilage out there, but what I have seen is that, yeah, if you do it really, really early, that it will regenerate.
And if it'll regenerate, does that mean that it can adapt? And I guess where I'm going with this question is it possible for, if you're sort of mechanically loading your cartilage during growth and development, are you going to have stronger, healthier cartilage when you're an adult?
Mm. Yeah, I think my guess would be it's kind of a double-edged sword, just because we know that, or that some of the literature that's out there in, I think it's dogs and mice, is that there is like thicker cartilage isn't always better cartilage. And plus when you're talking like a question of diffusion as well. And I know, and so in the PRG4 knockout mice. So those are the lubricin deficient mice. They're born just fine, and they will get for the first I think it's about 10 to 20 weeks of life, like they will have thicker cartilage than a normal wild type mouse. But then that cartilage breaks down like very quickly after, like there's this, thicker, and then like just this magical time point, it just like kind of crumbles and you get full thickness fissures everywhere. So and we're just doing the biomechanical testing on those guys now. But I think I can't remember, like, well, actually maybe was it softer? I think it was.
Oh, in the PRG4?
Yeah, I think it might have been softer.
Yeah, it was soft.
Okay. Thicker and softer, yeah.
I mean, I can keep going. What brings the stem cells to the location of the defect? Like, why do they go there?
Yeah, so our guess was, we thought it was going to be that CCL2, CCR2 pathway. But then, yeah, when we knocked it out, two things. When we knocked it out, we didn't see anything going into the defect anyways, but then when we didn't knock it out, we didn't see anything really going into the defect either. So we're not sure. In the C57 black 6 mice, it very well might be that pathway, but in the P21 knockout, it doesn't look like actually, it's possible that it could be as well, but jury is still out on that one. I think my guess is I'm pretty sure it still has something to do about that pathway, but I don't think we can address it by a simple like knockout. It's probably a gradient of CCL2 and if we just get rid of it completely, we lose the effect. We need like some magical level of CCL2 that recruits the cells.
But do you just have are there so many stem cells floating around in the synovium that they're on every surface, and if you have a defect, they're going to sort of bind to that surface and you're going to be able to see them? Or is something literally around the defect attracting them to that location? So
in mice, other groups, so like Cosmo Dabari's group in, is it Aberdeen or Edinburgh? I think he's one of the two. I think he might be Edinburgh. They've shown that these synovial stem cells can also home to the defect. They weren't able to show what the molecule was with homing either, so the jury, like I said, we're still kind of lost on that one. But Hunsinger in the late 90s, early 2000s, he did some experiments with rabbits. And like I was saying, rabbits are like this, they're this freaky one-off animal that they're just a good size for orthopedics, but they're their own thing, and they're not related to anything else on the face of the earth. Maybe octopus, I don't know, because nothing else is related to octopus. If you do a full thickness cartilage injury in rabbits, you'll see this really weird, in some of the rabbits, you'll see this really weird response where the synovium starts to proliferate, and then the synovium actually crawls as a caterpillar, like a piece of the synovium will start to migrate off the synovium and literally crawl into the defect of that cartilage defect, and then hive off, and then turn into new cartilage. And they haven't found another animal that actually does that. And a lot of people question that work because they can't find anything else that is even remotely similar. But yeah, like I said, rabbits are just, yeah, they're different. But it just shows that like this like kind of train of synovium can actually, they've got beautiful histological images of it, like just a piece coming off the main synovium going into that defect and then further later on differentiating into cartilage.
Yeah. Final goal is be able to repair cartilage.
Yes.
Will you get there? And when? And how?
Yeah. So this was just all on this presentation here is on the endogenous repair part of the lab. We also have a couple people working on the exogenous repair. We use synovial stem cells in humans because that's where our expertise lies. And that we also think that if there is going to be cartilage repair, it's the most likely candidates are the stem cells that are in the neighborhood. So what we found in humans, so we work with previously with Cy Frank, but now with Nick Motati and Alex Rozantsev. And so we collect synovial biopsies from normal joints, people that have ACL deficient people, and also people with full-blown osteoarthritis. And we take parts of their synovium, digest it, and then collect all the stem cells that we can collect. But collect those stem cells one at a time. Right? So there's one, we grow them all clonally. So it's like one stem cell from this patient in a dish. You grow it up till you get enough to do something with. It's a long answer, sorry. And what we do is we take all of those different stem cells when they grow up enough, and we look for their ability to turn into cartilage or bone or fat. But then we also take those stem cells and inject them into mice or rats that have that cartilage defect. And we see, even from the same patient, so let's say if we're able to grow 10 or 20 different stem cells from one patient, and if that's a normal person or an ACL injured person, an OA person, a couple of their stem cells will actually go to the defect, make new cartilage, and become new cartilage. We can see that cartilage is derived from those stem cells. Some of those stem cells go to the synovium and start pumping out a lot of pro-inflammatory factors, really, and the whole joint becomes inflamed. Some of those stem cells, we have no idea where they go or what they do, but you can't find them again, so maybe they just die. But the really, really interesting thing is that we find all of those types of stem cells in everybody's joint. So no matter if you're an OA patient, a normal person, or normal joint, or somebody with an ACL injury, all those types are there. It's the ratio that's always different. So in a normal joint, the vast majority of the stem cells will become new cartilage and actually fix cartilage defects. Even better than we see with these transgenic mice, they will become brand new articular chondrocytes. But there's about 60 to 80% of those are in a normal joint. In a OA joint, it's flipped. So you find less than 20% of those stem cells are still present in an OA joint. And in a ACL injured joint, it's like 40, 60. So they're just like, there's kind of this switch. And right now what we're trying to do is just find markers that can identify those ones that actually make brand new cartilage, so we can just isolate those and get rid of the rest, because the most like powerful result that we've seen is that, so if we take all those clonal cells, you take one at a time, you inject them, you get cells that, good cells that make cartilage, bad cells that make inflammation, and some other cells you don't know what they do. If you start mixing those cells together, if you take a good cell and mix it with an inflammatory cell, then the ability to make new cartilage is gone. That animal does not regenerate cartilage, does not repair. And so every stem cell trial on the face of the earth, like today, does exactly that. They just take all the stem cells they can get and inject them into a joint or inject them wherever they're injecting them to. And that's why we think that, like, when you have a clinical trial of 10 or 20 patients, because they're expensive to do, so they're small studies, you never show significance because you get 2 or 3 that show like this huge beneficial effect, 10 to 13 that show like, no real effect, positive or negative, and then a couple that show they actually got worse. And so when you put them all together, you get nothing. So if the Arthritis Society or CIHR gives me a couple $100,000, then maybe within a couple of years we'll be able to identify that marker where we can actually say this marker is expressed only on the stem cells that regenerate new cartilage, and therefore we can, when we're doing a stem cell therapy, we're only going to take those cells. So sorry, long answer.
Does it only depend on the stem cells that you're injecting into the joint? Is also the environment of the joint that you're injecting stem cells into?
Yeah, so that is completely agree. So one of the big limitations of the human work that we were talking about is that we have to do that in immunocompromised mice, because if you start injecting human cells into animals, 9 times out of 10 they're going to be rejected. So the animals that we are doing this in don't have a fully competent immune system. And before if we ever got to the point where we were going into clinical trials, we would have to figure out a way to inject those cells into a larger animal model that did have a competent immune system. Because yes, if you are all the clinical trials that have been done in advanced OA. So patients with advanced OA, these are patients with Calvin-Lawrence grade 3, 4, where they are months or years away from a joint replacement. Stem cells never fix their joint. You might see a beneficial effect of a couple months of reduced pain or something like that, but you never regenerated the joint. That I think they're not like this magical panacea or magic bullet that they can overwrite everything and do whatever we want them to. I think if you put them in an environment that's so hostile or so downhill that they're not going to regrow you a new joint, and if they did, that would just probably be called cancer. Yeah.
Building off that, so how do you kind of envision them when you actually detect OA, or how are you going to actually detect OA at a stage that would be beneficial to actually use stem cells, or is that possible or is it not possible?
Yeah, so we've tried to look, that's a part of my program for a couple of years there was looking at trying to figure out some early inflammatory changes that we could potentially cue on to be when this marker is down or something, this is the time you'd want to give somebody a shot of cells. And that's where we were collaborating with Carolyn and Jackie on their project. And yes, there very well might be, and circumstantially there might be a window. We looked at the inflammatory profiles of their 100, and there does appear to be a window that where inflammation is relatively it's not there's not a lot of inflammation going on, but the only way to know for sure would be to actually try it. Yeah.
But just what I was kind of curious is that will people be at a point when they come in, versus generally when they have pain, generally when they come in to deal with OA? Is that too late or no?
Yeah, if we were, let's say if I was going to propose to write a grant for a clinical trial, the first step I would focus on the injured, the post-injury population, just because you've got a defined time point of when you've got a pretty good guess of when this would have started, and then you've got there's tons of information that talk about inflammation post ACL, so we can kind of get an idea of when would be within the first 3 to 6 months probably would be wouldn't be a great time to start injecting these in, but yeah, that's if I was going to write a grant for a clinical trial, I would just focus on that one population. But again, that's not everybody that gets OA. You're not considering metabolic or just all those other risk factors that could go into it. Yeah.
I have a question.
Yep.
So the injury that you focus on the model, both the cartilage and the bone.
Yes.
I wonder if you just remove the cartilage without touching the bone, what would be the effect?
Yeah, so if you remove the cartilage without damaging or just even poking the bone at all, you get no repair. And that's in the MRL mouse, that's in the P21 knockout mouse. The reason that nobody's proved that like factually, but the reason that everybody believes that's the case is because there's really no like since there's no innervation or really no blood supply in the cartilage, it doesn't know that it's actually been injured. It's not the best. It's not the very scientific answer, but that's in all of these models that can regenerate. You see the same thing. You don't have to like, go and like, fully compromise the marrow cavity or anything, but you have to nick the bone to get a repair response. Yeah.
So it's interesting that you talk about that when the cartilage is regenerated, no stem cells is in that repair tissue, and you are saying that maybe the cells from the adjacent area is migrating into the repair tissue. Have you looked into the density of the cells to see if the density of the cells decrease for the repair?
Yep. So right now we're actually doing we're labeling the chondrocytes. So fluorescently labeling the chondrocytes and doing the same injury to see if now it's the cells that. So if you label all the chondrocytes before the injury, do the injury, and then you get a new piece of cartilage that's made of chondrocytes, you know that it was like the existing chondrocytes that then repopulated the area. It's just that sounds easy, but it's really a lot of complex genetics that it's actually really tricky to address that one, but we are actually doing that, or trying to do it anyways. We do think that chondrocytes are re-entering the cell cycle and proliferating and migrating. We just have to prove it. We're running out of cell types to test. So that's one nice thing about cartilage is you don't have it unlimited. There's only got like 4 choices of what can be making the new tissue. So we've knocked out like 2 or 3 of them. So yeah. A lot of negative results.
These are, you think of your existing osteochondrocytes, not...
Yeah. The one piece of evidence that we do have that's very strong, sorry, talking too loud into that, is in mice long bones, it's absolutely agreed upon that PRX positive stem cells make bone. Like there's 50, 60 studies that show that the same thing, that different places in the body, PRX positive stem cells make bone. In that defect, we never see the subchondral bone being made of the PRX positive cells. We see them hanging out there, but they never make the new bone. So there's got to be another cell type that is making that new cartilage and another cell type that's making the bone. And we thought we'd at least have that to fall back on, but it's never made of the PRX positive cells.
So that reminded me of something you said during the talk, and you said that you've observed that the cartilage grew from the outside
in.
And I was like, so I was going to ask you, would you have expected something different?
I would have thought it was going to like start as little islands and spread outward from those, but like that was just a guess. Like nobody's done that experiment yet because very few things heal articular cartilage. I would have expected, especially if it was stem cell mediated, that you would get stem cells migrating, attaching to the bone, and then they would form a little colony and then make a little island, and then there'd be another stem cell, make a little island, and eventually all those islands would join up. But that result there also makes me think it's the chondrocytes that are just kind of coming over, yeah.
Because it also, the existing cartilage provides like a scaffold of some kind for them to actually
but there is no
build something off.
But off like the existing ones that they're climbing out of, right? But that's what we think the stem cells are actually doing, is they line up on the periphery. And if we actually isolated those stem cells like from the mice when they're like from the defect, from like the joint, and those stem cells, the P21 stem cells, are pumping out a ton of extracellular matrix that is like cartilage related. The C57 black 6 stem cells are actually pumping out a ton of matrix that is bone related. So it's all it's all kind of collagen 1 based matrix. Yeah.
All right, any other question? Thanks.
Oh, thank you all.